Malaria is sort of, I would say, one of the most deadly diseases after HIV and tuberculosis. And it sort of kills almost 1 million people a year. And I think every 32nd there is a child dying in Africa. I'm Dr. Rita Tiwari, and I'm working at University of Nottingham in School of Biology, and my main area of research is to understand malaria parasite development. And so the area what we work on is to understand what are the different signals needed for parasite to develop. The disease is really caused in the human, where they sort of normally attack the red blood cells, and that's how people get very high fever. But there is other part to this parasite, which is within the mosquito, that bites and causes that disease. And that part of the life cycle is not so well understood. And so what we try to understand is what happens within the mosquito, and if we can control that, then we can sort of block the transmission of this parasite. So it's not only the disease itself, but it's also the transmission, which is important to control, because if we want to eradicate at one stage a parasite from this world, we have to sort of eradicate the transmission of malaria parasite. Vaccination do not work fully. We have only 30% of the people in Africa which are protected with the vaccine that is present now. But the problem with the African continent and also the Indian subcontinent is that they take drugs which is to kill the parasite at the blood stage, but there's lots of parasites which are resistant to these drugs. And so we need new drugs, we need new vaccines in order to control the disease in the human being, but we also need somehow that the disease doesn't spread. And that is the major problem. So we work on certain families of proteins which could be very good drug targets, and these are called kinases. And these have been a drug target in cancer and many other diseases. But in Plasmodium it has not been, or in malaria it has not been explored so much. And so what we have done here is to sort of look at all the kinases which are there, which are regulating or controlling the parasite development, and to find out which one could be good drug targets, which could be used to sort of block either the parasite within the human host or to block the transmission. And so that's why the work is important, because this is the first time we have sort of studied all the different kinases in any system. And since malaria parasite had something like 72 of these kinases, we have tried to look each one of them by taking away from the parasite to see what it does to the parasite. And we show that how some of them are important for the transmission, and that is the power of this screen that we have done. So yes, so this life cycle is very complex. So it goes one in the human host, or the mammalian host we call it, and the other one is the vector, which is the mosquito. And the real disease is in the human host, where it sort of invades the red blood cells, and it sort of produces millions of these parasites. And that's why people get very feverish, get chills. And then when we give them treatment like chloroquine or artemisinin, the parasite population goes down and they are treated. But there is another stage of parasite which are called sexual stages, or you can say male or female stages of these parasites, which resides in the human host. But when the mosquito, which is not infected, picks up these, it's within the mosquito that these sexual stages then go through fertilization, the sperms are formed, and then they fertilize the female, and we get further development of this parasite. And this is what is the transmission stage. So if we can block that sexual stages, then the transmission doesn't happen. Now these stages within the mosquitoes can develop further, produce millions of sporozoites, which is another stage, in the salivary glands, and when the mosquito bites, the infected mosquito bites, it again gives rise to these parasites in the human host. And that parasite is dormant, so we don't see it for 48 hours when they infect the liver cells. And that's another stage where people want to block it, where the transmission can be blocked. If we can block at the liver stage, then the people will not get the big disease which kills people. So there are three main stages. One is in the blood stage, one is in the mosquito stage, and one in the liver stage, where they produce millions of these parasites. And that's why it is difficult to control them.